Hello everyone, welcome to another discussion on our series on neuroscience and neural networks. A quick recap of the discussion we've had so far. We started off with a basic premise that yes, there are actions that we take on a communal basis based on traditions and very deep-rooted customs. However, when thought about those customs, those traditions on an individual level, we may find them to be morally invalid. Why do we consider them communally very much accepted in our society? We found some reasoning based on that. It's because of the way our brains are made to think about them as very normal. We included bribery, corruption, dowry, and many other social evils in that. However, we tried to understand, why is our brain wired in the way it is? Why do we think the way we do? To that end, we wanted to understand certain behaviors, and we wanted to develop some reasoning with respect to the neuroscientific elements associated with those behaviors. So we wanted to understand neuroscience, began with its definition as a study of the nervous system. For that, we looked into the structure of the nervous system, and we figured out it's constituted of the central nervous system, the peripheral nervous system, and within those there are different elements and functionalities of certain parts, and we looked at those in detail. Then we wanted to look into the building block of the nervous system, and that happened to be the neuron itself. So we tried to understand neuron and its functions, and began with our discussion on its basic structure. Figured out they are dendrites, tree-like, thread-like structures that are basically there to receive information. There's a cell body consisting of the nucleus, some different organelles within the cell body, and then we identified a channel of communication, the axon, which is where the electrical activity takes place, the information transfer takes place, information is basically carried within this axon. That electrical impulse is then transferred to another neuron via the axon terminals towards the end. So that was the basic structure of the neuron, and that axon channel we figured was covered by a protective myelin sheath meant for insulation and protective purposes. Schwann cells produce this myelin in the peripheral nervous system, and oligodendrocytes produce this in the central nervous system. In our discussion of the neuron, we further differentiated the neuron into different types, and those were based on functional types or the structural types. So structurally, we divided the neuron into different types, such as the unipolar type of neuron, the bipolar, the unipolar, the bipolar, the multipolar, and the pseudo-unipolar types of neurons. Okay, and if I were to show you another representation of this, and we said the naming convention was fundamentally based on the number of passages coming out of the main cell body. So the unipolar had a single passage, the bipolar had two passages moving out of the cell body, one for the dendrites, one for the axons here. You have a dendrite and axon on the unipolar as well, but there's a single channel moving out of the main cell body. Here you see two channels moving out of the main cell body, whereas in the multipolar case you have multiple channels coming out of the main cell body. Okay, so based on that we defined different types of structures of the neuron. Then there was a functional differentiation as well, and this functional differentiation was the sensory, motor, and the interneuron types of the neurons. Okay, and we figured out that each of them have a different function of their own. The sensory neurons are called the sensory neurons because their main function is to sense different stimuli, receive different information, and then move that on. The motor neurons, and these are typically these are unipolar in nature. Okay, and that also makes sense. I'm going to leave that as an exercise for the viewers to figure out why are these typically unipolar in nature. The motor neurons, as the name suggests, they are the kind of neurons that are going to have the final impact of contraction, relaxation of skeletal muscles or glands, for instance. So, and these are most of the times these are multipolar in nature. Again, the viewers are encouraged to figure out the reasoning uh, as to why these are typically, almost always, they are multipolar in nature. And then the interneurons, which form the bulk of the neurons in the nervous system. These are also now they are typically multipolar in nature, but they perform a connective role between the sensory and the motor neurons most of the times. So that was our bit on the overall recap for today. Our discussion today is fundamentally on the neuronal activity that takes place, the electrical activity that takes place in the axon. Okay, so how does the electrical signal move within the axon? What exactly happens? What exactly triggers it? How does it move on? What are the different stages in those, um, in that activity specifically right here? Okay, so we're going to see what that is. So that brings us to the concept of action potential. Okay, we begin this by understanding that there's a resting state for all neurons. There's a resting state at which the neuron has a particular electric potential. Okay, and how is this set up in negative? That is the question, first of all. So we recognize that each neuron has a cell membrane, and those cell membranes allows or it regulates different ions to move in and out of the cell. It so happens that most of the times you have a majority of sodium ions set up outside of the cell, and the potassium ions are set up within the cell. There are other charged ions as well, chloride ions. There are certain proteins and certain ions associated with those proteins, certain ATPase ions as well. Uh, and as a result, the inside of the neuron is relatively negative in potential as compared to the outside of the neuron. So there are more negative. It's safe to say that they may be both of them may be positive in nature, but the outside of the cell is more positive as compared to the inside. Okay, so that's one way to look at it. That although you have potassium ions on the inside, you have sodium ions on the outside. They are both positive, but it's safe to say that it's more positive on the outside as compared to the inside, resulting in a negative potential on the inside. Okay, just a quick example. If you have 5 volts on the outside, 3 volts on the inside, it's safe to say that the inside is at a relatively negative potential to the outside, even though it's positive. Okay, so that being said, just a crude example, that being said, the inside of the neuron is at minus 70 millivolts. This is a rough estimate. This is a rough approximate, and that's what the inside of the neuron is at the resting potential. Okay, what happens is when the neuron encounters an external stimuli, when it faces some external pressure, or chemical stimuli, or any external stimulus, basically, that it could be electrical in nature, it could be some pressure. Pain is one stimuli that almost all neurons face. So any external stimuli is going to generate an electrical activity, or it's going to cause some sort of a potential difference within the cell, within the neuron. We run with the idea that if that stimulus is stronger such that it causes the action potential, it causes the electrical difference within the neuron to be greater than minus 55, or any threshold value, minus 55 in this case. If it's higher than a threshold value, if that action stimuli is such that it causes the potential difference to rise beyond the threshold, the neuron is said to have fired. The action potential is said to have achieved. Anything less than that does not result in an action potential. Okay, so the idea is that an action potential either happens or it does not happen. Once you have met the threshold, the action potential is guaranteed to happen. Okay, so that being said, uh, what exactly happens when you look at the neuron during the action potential? Why are we seeing this happening? Why are we seeing the action? Why are we seeing the electric potential changing so rapidly? And this is in fact action potential in itself. It's a brief reversal, as you can see. There's positive on the upper side, there's negative on the lower side. So it's basically a brief reversal of the resting membrane potential. Okay, the membrane potential is at negative, and then there's a brief reversal in that. It could also define it as the explosion of electrical activity and impulse by any depolarizing current, okay, or the movement of ions for that matter. Uh, if I were to talk about different stages, you could see there are different stages set up. There's depolarization, there's repolarization, and then you go back to the resting state. Okay, so we're looking at these stages, and let's identify them one by one to see what happens in each of them. You can see there's a depolarization state first off. The depolarization state, what we are basically looking at is a rise in potential, yes, and that is attributed to the sodium ions that are greater in number on the outside of the membrane. These sodium ions, there's a strong influx of these sodium ions into the cell, into the neuron. And once these sodium ions move into the neuron, the inner side of the neuron is at a negative potential. Okay, it's at a negative potential. Once these positive sodium ions move in, what you experience basically is these positive ions moving into the cell and negating the already negative inner potential of the neuron. So these positive ions literally depolarize the cell, okay, because they cancel out the polarity. There's a negative potential that has already been set up. These positive ions are coming in to negate it. Now if it reaches at zero right here, then we talk about the depolarization being very much perfect, okay, but that's not the case. What exactly happens is the sodium ions move in, um, let's just say sodium channel opens, sodium ions move in, such that there is depolarization, but then there's even more depolarization. There are more of these sodium ions moving in, such that you reach a positive um, relative potential inside this neuron. Okay, we're talking about inside the neuron. So this graph is for the inside of the neuron. The inside of the neuron was at a negative potential. The sodium ions moved in. They negated that negative potential. Even more sodium ions moved in, and that resulted in a positive potential inside the cell. Now, to counteract this influx of positive ions, the potassium channels open up and they allow the potassium to move out. Okay, so the sodium ions are moving in. When excess of these sodium ions move in, to counteract that influx of sodium ions, the potassium ions, they're also positive, they move out. So these positive ions moving out, leaving the cell, results in the decrease just one moment, let me find the right color for this. Okay, so these potassium ions, moving out of the cell. Potassium channels open up and they decrease the potential within the cell. The exact terminology for that is repolarization, because they are decreasing the inner potential of the cell, of the neuron, by moving out. Okay, so these potassium ions move out, they repolarize the inside of the cell, okay, or this the membrane. So once that happens, it so happens that it goes even beyond uh, the original resting state. Okay, so once again, the first we saw the axis of sodium influx, and then we're seeing an axis of potassium outflux from the membrane, from the inside of the cell. Okay, so that results in the cell being way too negatively charged. Okay, the potential difference within the cell becomes way too negative. What happens then is there is another corrective measure that takes place, which is called the refractory period. Okay, the first thing that you must intuitively figure out in this refractory period is that the potassium channels need to close. Okay, let me also clarify, we forgot to close the sodium channels right here at this point. Sodium channels close here. Okay, so the sodium channels, they closed there and then, and the potassium channels open. At this point in time, the repolarization started taking place. Once the repolarization, there was excessive repolarization, the potassium channels closed up, and then a corrective measure, a refractory period, started off. And the refractory period basically leads the cell back to the resting state. So very intuitive to understand, the refractory period is going to allow the potassium ions that went out of the cell to come back in, and the sodium ions that came in the cell to move back to the outside. Okay, so that is going to bring the cell back to the resting potential. So this is basically a corrective measure. It's an opposite of what happened initially. In the depolarization state, we saw the sodium ions move in the cell. In the refractory period, you want those sodium ions to move back on the outside to get to the initial state. In the repolarization state, we saw the potassium ions moving out of the cell. In the refractory period, we expect them to come back into the cell. Okay, and this is regulated by the sodium and potassium pumps set up in the cell membrane. And it is said that for each 3 sodium ions moving across the membrane, there are 2 potassium ions that move across the membrane. Okay, so that's which also makes sense, because if the number is not equal, and which makes sense because the number is not equal, and that inequality of the number is basically leading to a considerable voltage difference inside the cell as well. So this is what's happening, and this is what's happening in the action potential in itself. But also important to understand that this is happening at one instance. This is a very localized phenomena. It's happening at one instant. You have an axon hillock right here. That's where it starts off, and you expect this phenomena to happen right there and then. Okay, if you closely look into this diagram right here, you expect one stage of the action potential to be taking place. For example, there's depolarization taking place here, and then the next part of the axon, the depolarization follows in the next part. And while the depolarization follows in the next part, there's a repolarization in the previous part. Okay, and that's how the action potential, the spike that we see here, is, remember how there's a spike in the depolarization and repolarization. There's a spike right here between these 2 stages. So when we move here, if I were to draw right here, so there's a spike right here, and we expect this spike to be between the depolarization and the repolarization state. In fact, this spike was right here, because after this depolarization there was repolarization, so there was an electrical spike right here, and then the same happened here, and then the same happened here, because repolarization is following a depolarization. Okay, so and then obviously there's a refractory period and resting state. So all four stages come through one part of the axon, and then all four stages move to the next part of the axon, and then the next four, and then the next part of the axon. But they're happening sequentially, so they're not happening simultaneously at one point in time, which is why we say the electrical signal moves through the axon. So while one part of the axon is experiencing one stage, the previous part is following the previous stage. Okay, this happens sequentially, and that's how the electrical signal moves through the axon, reaches the axon terminals, and then the information is transferred to the other neuron. So in this video we looked into the intraneuronal communication, transfer of signals. In the next video we look into what happens to the electrical activity. How is the information being transferred from the axon terminal to another dendrite or another neuron? Is there a chemical interchange or an electrical interchange? Are there any neurotransmitters that are making that changes? Here we saw there's a purely electrical transmission, but it is regulated by their voltage pumps and voltage gated ions in the membranes. Okay, thank you so much.